Okay, everyone. Welcome tonight for the WARACI Camaraderie for June. It's good to see everyone around today. We've got two events running simultaneously with the titration competition going on upstairs and this one here. So, yeah, it's a good amount of people getting involved. I think there's 27 teams involved upstairs. So, yeah, it's a good turnout. So, my name's Terry Humphreys and I'm from Curtin University and I'm your host for this evening. And I'd like to start by acknowledging the traditional custodians of the land we're meeting on today, the Wujek people, and I wish to acknowledge and pay respect to their continuing culture and contribution they make to the life of this city and the region. I'd also like to thank the sponsor of the Chemaraderies series, which is Chem Centre. Without those guys, we won't be able to have these refreshments before and after the event, so thank you to them as well. Just a couple of future events that we've got coming up. We've got the What's New in Laboratory Instrumentation Expo, which is this Friday. And so I think registrations have closed. But yes, it's going to be an excellent event.
You can probably register, additionally register, because I'm in charge of it. So.
Okay, so if you want to get registered, speak to Peter. He can help you out there. So it's a fantastic chance to come down, meet a lot of the instrument manufacturers, producers, retailers, and come and see what's new in the laboratory scene. So next Chemaraderie is going to be in July, and that's going to be on the 22nd. I'd originally said it's going to be the 17th, but we've had to move it. And so Associate Professor Dino Spagnoli is going to be here to talk on molecular solids on Titan, a moon of Saturn. So the registration page for that will be open pretty soon, so hope you guys can attend. So on to the main event. The reason why we're all here is for Dr. Toff Gilliam, and he's going to be, he's come to us from Murdoch University, and he's going to be talking about navigating the labyrinth of early career chemistry side quests. And so I'm not going to give a big bio on him because he's going to be telling us all about his journey. So please welcome to the stage, Todd. Okay. I think that should work.
Cheers. So I had to think of a fun title on the spot, and that's what happened. So I've had a bit of a weird working life. I didn't actually think I would likely ever go to university. In fact, my careers counsellor told me that I might be able to get a job at Blockbuster. I did my work experience at Blockbuster. They didn't like me because my pants were too long or my boxers were too high. So when I left school after generally underperforming in all areas except for visual arts, I became a general tradesperson and builder's assistant, and then started doing air conditioning and sort of general handyman work for a while. And then I started crawling around in a roof space in summer working for a small company that didn't really care if it was 42 degrees. And I thought, this sucks, and just applied for adult entry to university and didn't understand anything about ranking systems or what universities were, and went to my first preference, which was a Bachelor of Science at UniSA, which I thought was down the road, and I was wrong, and I had to go into the city for the first time because I never bothered catching a bus that way. So I went there, and it was a really good time. Don't know how to work PowerPoint still, but you know, we're doing it on the fly. Oh, do you know why? Because I don't have my presentation open. Again, as I said, educational deficits. So I went to university to become a biologist, and I ended up dual majoring in chemistry. And come the end of it, I really wanted to do entomology. And the guy that I really looked up to in entomology had a massive falling out with the rest of the department and quit. And he was like, you're still pretty good at chemistry. You should probably just do that. So I joined this sort of med-chem drug discovery group. So it was like this mega group at UniSA. And after a year of making many, many derivatives of anti-cancer compounds, I got really sick of seeing how many fluorines I can put on the same scaffold. And I joined the team of Sally Plush and started looking into metal ion complexes for cell imaging applications. So this was an opportunity to try and marry my sort of passion for biology with synthetic chemistry, which I started to really enjoy. This was in collaboration with Max Massey, who many of you would know. So he was my co-supervisor over this end of the Nullarbor, and started working away on these rhenium complexes as well as some iridium and terbium complexes that were just way less impressive. Not that this particularly achieved anything, but my project was about bioconjugation, starting to put biomolecules on these complexes under the concept that these metabolic motifs might have some sort of difference in behavior when they get taken up by cells, and that might label things that are different in a cancer or disease state. So the good cover slide for that is these are normal cells and these are disease cells, and they kind of look different. But the benefit to metal ion complexes for luminescent imaging is that they persist in their phosphorescent emission or mixed luminescence for a particularly longer period of time, and you're able to gate out autofluorescence in the background. It gives you a sort of better idea of what you're actually looking at, and it's not contaminated by endogenous fluorophores. So at the time, Kenneth Lowe was particularly hot in this field with looking at the small sugars and chucking them on there, and they're going into different regions. And a lot of his complexes tend to accumulate in the mitochondria. It didn't really matter what complexes people were working with, they're always going into the mitochondria. And people are like, well, mitochondria is powerhouse of the cell, and it uses sugars, and that's why the sugar's there. But it turns out that these are actually just large lipophilic cationic species. So they tend to cross mitochondrial membranes in a direction which they will accumulate there. And as a result, they're very toxic. So Max and his team started developing these charge neutral complexes, negating the charge on the rhenium, and they're sort of accumulating in different things. So these are still quite lipophilic, and they're labeling lipid compartments in cells. So we thought, what if we took that and started chucking some sugars on that and see what the difference is? So the benefit was that these are easily excited, give a long-lived emission, and they exhibit a reduced cytotoxicity because they're not disrupting mitochondrial membrane potential. And they feature this click conjugation site where we can rapidly just chuck stuff on them. And in the words of Sally Plush, suck it and see, was the approach for her research group. And she said that weirdly often. So in that regard, took a bunch of sugars, seemingly randomly chosen in retrospect, but you do trust the wisdom of the people around you at the time. So we chose glucose, galactose, mannose, and maltose, and we tried to choose their representative anima, or the position of the C1 carbon can project in either an alpha or beta orientation. So we picked that which was most represented biologically and produced these azide click conjugates and whacked them onto the rhenium alkyne complex. They all exhibit the same properties in terms of fluorometrics, and they are sort of differing only in their lipophilicity, but they also fit no trend of lipophilicity for their relative cell uptake. So we do think that there might have been some contribution of an active cellular uptake process associated with this, and we're looking at this in terms of prostate cancer cells, because Sally's group sort of joined with this larger prostate cancer research group at the time, and they were developing the company Resolve, which some of you may or may not recall from back in the day. Resolve, I don't think, worked out, but basically, working with the cell biologists were saying, oh, you know, there's more rhenium mannose in some of these cells. You can't really rely on what looks like more or less. So we did some ICP measurements and determined the relative cell uptake in these. What was interesting is that we started to realize that rhenium mannose in particular had this very nice accumulation in small compartments within the cells. And we associated that with the potential deregulation of liposomal handling within prostate cancer cell lines. So when we started to compare across these and we were trying to determine whether we could, inhibit their uptake. Couldn't. But we started to see these sort of labeling of small extracellular compartments. I have really shaky hands, it's probably heavy metals. But in any case, the difference between sort of normal cells or transformed cells was the accumulation of the complexes within these small compartments that tend to get trafficked onto the outside. And we were hoping that this might indicate some deregulation in liposomal handling associated with the disease state. As I mentioned earlier, the benefit to these is they persist and they can emit for a long time. They don't tend to degrade like a lot of organic fluorophores, although there have been advancements in that since. And they sort of accumulate in cells. These are human THP1 macrophages. You can see the increase in green signal showing the influx of the rhenium mannose complex. And the red on the outside is an organic fluorophore called cell mask, or labels the plasma membrane. Because you're constantly exposing this over 30 minutes, the fluorescence from that is depleting and the metal complexes persist. So they're good in that regard. And Max's team have gone on to develop a wide range of these that tend to be even better still. So that was kind of the end of that journey. And come the end of my PhD, I decided that I'll just do another completely different thing. And I joined a polymer research group. So I joined the Applied Chemistry and Translational Biomaterials group, mostly because I had taught most of their students throughout undergrad. And it was just a team of people I liked. So I applied for this job with Anton here, and we took on a bunch of PhD students during the time that I was there. Ala makes sort of drug delivery systems for injections into eyes. Manisha here is still going. She's a bioconjugate chemist. She looks at new mechanisms and ways of bioconjugation. Saddam's just finished. He is working on nanoparticles for drug delivery in ovarian cancer. Ashlyn Austin studies the smell of sheep and then formulates them into baits to attract foxes to not quite poison them, but give them something that doesn't taste nice to try and control their behaviour. And Cynthia here also worked on pH-responsive basically micelles that were going to disassemble at endosomal pH in the attempt to try and release cargo. So it was a bit of a mixed bag of a research group. Not everyone's represented here. And suddenly they had like a year's funding. And Anton's like, I don't really mind what you do, just do something. So I started making these plasma polymers to coat, you could use them for implants or coat any material. These had the advantage of, at the time we were looking at nanoparticles, I was reading about silver nanoparticles and they seemed really cool. But they're somewhat limited if you're trying to coat different materials like ceramics, titanium, bandages. You have certain limitations to how you're actually able to bond these things unless you just saw surface coating. So we realized that triiodides or iodine generally is quite good at penetrating biofilms, and it has to be stabilized in a way that can be secreted long term. So many of you have probably used betadine. So it's povidone iodine, which is just basically this polymer but in little globules that suspend in a solution. But instead we used a plasma reactor to cross-link these monomers in a way that was able to complex iodine as a stable triiodide species. And then in a wet environment, such as a burn or wound or an implant, it slowly releases the iodine and can have an antiseptic effect. So the benefit here is that you get broad spectrum antiseptic action that can penetrate biofilms. Somewhat better than some of the other platforms for doing this. There's been some follow-up work on this still. But at that time, I was working on some rotaxanes, all sorts of weird different things, trying to form templated crown ethers. It was just kind of like Willy Wonka's chocolate factory of mystery chemistry. So this one was a success. We tried many things that weren't, and largely my job was sort of coordinating the lab and training up students. So it started to look a bit dicey, and I was about 3/4 the way through my contract, and it didn't look like there was any more money on the horizon. And Anton and I joined this PRIF mining consortium. So the PRIF is the Premier's Research Innovation Fund. And he's like, do you reckon you could do minerals research? And I was like, I reckon I could do minerals research. So I joined the Minerals Research Consortium between Adelaide and UniSA and joined this PhysChem lab. So at that point, they gave me this like range of projects, and I was like, oh, RP7, I've used that before. No, I got given this flotation project, and I was like, what's flotation? So I Googled it, and flotation is a really good way of separating high-grade ore from low-grade gangue or crappy ore. And you use physical chemistry processes to attach small ore particles to bubbles. They float up, you skim them off, and you collect them. So I recorded this live conversation from BHP's Olympic Dam, where these two workers, they go, oh, how's the froth looking? And they go, yeah, it's pretty good. And they're not really sure how good it is because, as Kathy Ehrig, who oversees superintendent at Olympic Dam, said, there's not enough grey hairs left in the mining industry, and now we're trying to measure things with fancy instruments, but you just need to look at it and know if it's good froth. So that was my understanding of, that was the best way of determining whether there are enough surfactants present to achieve a good froth. So it's kind of like a large boundary of error there. So yeah, they're like, just chuck more in. So they have this issue where they spend a lot of money on frothing agents and they just keep dumping them in, but they don't really know when it's optimal. And if you dose too far, you lose profit because it doesn't become efficient. So froth flotation, so far as I understand it, was that you have a bunch of surfactant molecules, this is what it said on the bottle, that they stabilize a bunch of bubbles that are like agitated and frothed into a column. And then as those small bubbles move up through a slurry of ore particles, they are mediated by xanthate collectors, or it could be some other collectors that decorate the surface of the ore and interact with the hydrophobized bubble, rise to the top, they're skimmed off. So the question that was presented to me was, Todd, do you reckon you can measure these gravimetrically in solution using a quartz crystal microbalance? And I was like, what's a quartz crystal microbalance? And they said, it's a little crystal that shimmies back and forth, and when things touch it, it does it more slowly. And I went, yeah, I reckon I can do that, because I wanted to stay employed. And staying employed is a real challenge post-PhD. So I went, it'll probably be fine. So I went, okay, so I need to design something that might bind these or hold them. And they said, you've worked with polymers, because I'd done 6 months of working with polymers. So I was the polymer guy of UniSA at that point. And they said, we need polymers that are molecularly imprinted to bind to these things. But these things are really wibbly-wobbly and flexible, and they move around. And all the literature that they gave me as examples used really large, rigid, specific things that have functional groups and really specific orientations that it turns out you can template polymers to. And then they said, just test it on like a really rigid thing. And we made templated polymers that worked. And I said, I still don't think it works for wibbly-wobbly things. And it doesn't. So they said, well, you're employed now. Can you do something with it? And I said, yeah. And I played around with the quartz crystal microbalance for ages. Still don't really know how they work. But I decorated that with a bunch of polymers and cyclodextrins trying to bind these things in solution. And it turns out it's just way below the limit of detection. But because I've been making all these polymers, I thought, I'll just spin this differently. So I said, will you keep paying me if we just did something else? And they said yes, and it was fine. So it turns out that the frother that they buy that is meant to have 4 things in it has heaps of things in it. And they're all like generally the same. But if you want to produce something cheap, you're not going to like put nice, clean, expensive Sigma Aldrich compounds in there. So you get this like mixture of frother compounds or mixed reagents. So characterized them and went, they're all more or less samey. And when I say samey, they're all surfactants with hydrophobic tails. So I was, at the time, making these rotaxanes. So I was trying to thread cyclodextrins onto a long peg chain and cap them with photoreactive groups in a vain attempt to make these dynamic antimicrobial peptides that never went anywhere. But I had bought a lot of cyclodextrin. So with that lot of cyclodextrin, I thought it's really good at including anything into it. And I've spent heaps of time NMRing stuff that gets stuck in cyclodextrins. So the only problem with cyclodextrins are that they're really soluble in water and so are all the things that you're trying to measure. So I cross-linked them because I was the polymer guy now, and I made polymer particles. And by particles, I mean like rocks. So I took these rocks and I exposed these rocks to the frother solution, and I went to BHP and Magato, who were sponsoring me, and I said, what if we said that you've got a downstream process for cleaning the water that runs off from your process, and then you can recycle it? And they're like, that sounds good, say that. So instead, the project became stripping frothers from water and saying that that was the goal all along. So we found out that these polymers, as you dose them in, are particularly good at sequestering all of the surfactants, and that sets a precedence for like being able to repurpose water at different areas of the mine process site, because mining typically happens in areas where there's not a lot of water, and there's increasing demand on minerals processing groups and mining companies to reduce their water usage. So it actually turned out as a win for them. We also worked out a metal organic framework for sensing ferric iron and made like a polymer that was able to respond to that colorimetrically, but that was more of a collaborative work and less of a bumbling by me, so I haven't included it. Simultaneously, a lot of the students that I had with Anton and Hugo, who's a guy that was on my honours team when I was doing the medchem work. Anton's interested in polymer particles for drug delivery. Hugo's interested in how many kinase inhibitors you can get into one cell. And we were like, we're going to do this project where we identify receptors, make particles that are going to deliver drugs to them, and then put combinations of different receptor targeting groups and different drugs in the hope that we will have some sort of combined pincer maneuver on cells that we want to actually poison with chemotherapeutics and get fewer chemotherapeutics into healthy cells. So in that regard, we started doing some biochem stuff, and Varanga here and Ria are still continuing PhD students in that regard. So Ria is foremost a biochemist, and she came on to do a lot of the receptor screening and looking at different cell lines, RNA-seq data, and identifying different surface receptors associated with, at first, ovarian cancer, but then we included prostate and breast cancer cell lines, and then we stratified this into different groupings of ligand families and took on Varanga, and Varanga is now looking at bioconjugation of different ligands that are able to target those receptors, validating those and building the particles that are going to ideally go through to be tested. Now, that does have certain constraints and limitations. One of the issues is that particles are quite large and it's hard to get targeting vectors onto them that are going to work effectively. And if you do manage to get them into a cell, they typically end up in the lysosome, which is where I'm experienced with things ending up in. So our concept here is that if we were to target different G-coupled protein receptors that are co-overexpressed on particular cancer cell lines or cancers, we might be able to develop like a personalized approach to therapy. So a lot of cancers, such as ovarian cancer, are very heterogeneous in that they occur via so many different mechanisms, and it's very rare that people have sort of groups of cancer that are samey in terms of what you might be able to target. So the idea is that if you were able to sample that, run an analysis of what's being overexpressed, pick from your receptor targeting ligand library, This is more blue sky, but at that point you can sort of make a combined therapeutic vehicle that goes into the endosome and then releases that cargo. So in a pursuit to be able to, assuming that we can do that, you know, everyone's working on their bit, we want particles that are able to disassemble once they're in the endosome or lysosome. And we know that the endosomal, lysosomal environment is very acidic compartments within the cell, and we are assuming that once we have tubular local aggregation of the particles by the enhanced permeation retention effect, that we would then get active cellular uptake via these receptors, and then it goes into these acidic compartments and it disassembles. So Cynthia here, who's now finished, made a series of micelles and characterized them using polypyridal hydrophobic blocks for the assembly of these micelles that were able to respond to targeted pH environments. And this was reliant on the fact that they'd become protonated and charge repel from each other and dump whatever was inside. So Cynthia made many of these things and was able to demonstrate that, you know, across time, as you lower pH, they're sort of getting particle distributions of a known size. And then as soon as you cross into the lysosomal pH, you get disassembly of those micelles into unimers, and this is associated with the release of that payload or cargo. So Cynthia is now, talking of side quests, I told Cynthia to just go and do many things that she enjoyed doing her PhD, and this really annoyed Anton. Cynthia starred alongside George Clooney and Julia Roberts in a film during her PhD, because I encouraged her to go and do that instead of her work. And then she published a bunch of papers and decided that she hated academia and became a science communicator, which is pretty cool. So anyway, she started working with chemotherapeutics and demonstrating that we could have this, I won't say controlled release, because micelles are inherently leaky. They're kind of dynamically assembled at any given time. But we were able to slow the release for more of a burst after crossing over into the lower pH environment. And this was a good step in the right direction for this overall project. So that continues today. But now that I'm at Murdoch working with other groups, we're also looking at silica nanoparticles, liposomes, and micelles, each offering sort of different advantages. I don't know how I'm doing for time. It's rough, I'm rambling. No, we're good. We're at the somewhere mark. So during this time, I started stressing again because I was like, I don't know if I'm going to stay employed. So I wrote my first grant application, like fellowship application, and I had this conversation with Anton. He's like, oh, there's a National Intelligence Postdoctoral Fellow grant thing open, but you've got a really short amount of time to write a project and send it in. So his advice to me was put your conspiracy hat on and just write something that sounds alarming and then try and figure out a way to resolve it. So I did. So the most alarming thing that I found at the time was a lot of like media around poisoning with nerve agents, which on this graph that I nicked says that these are a lot worse than those, but they're all generally pretty bad. So the way that they work is that there are a bunch of typically volatile organophosphorus compounds that bind to acetylcholinesterase. Acetylcholinesterase is a little enzyme that moves around in your synaptic clefts for your neurological transduction of movement and breathing and all the things that you do involuntarily. And if administered, causes you to sort of spasm, all of your muscles go into lockdown, I suppose, because acetylcholinesterase can no longer take your neurotransmitters out of the receptors that are turning your muscular contractions on. So it's like a permanently on switch just being hit in your body. And so your eyes sort of contract, all your muscles contract, you go into a seizure and you produce heaps of mucus and then you drown in your own snot is essentially the way that you go. And they fall into these three classes. This was my conspiracy dive for a weekend. And I was like, these sound awful. And I was like, oh no, there's ways you can kind of work with safer representatives in a lab. We can do something here. And then the brief that they give you is very, very brief. And it says, we're concerned about the implications of nanomaterials for chemical, biological, and radiological threats. So I was like, oh, how can that enhance these? So I was like, if these are bad enough as it is, and people are trying to detect them, and they're easily transmitted and only requires a small amount to kill someone, what if you can't detect it? And what if they persist longer in the environment, or what if they can be aerosolized because someone's like wrapping them in a veneer or something? Because that's analogous to how we're trying to get drugs into things. And what would we do about it? So the basic premise is that you always need protection mechanisms, you need detection platforms, but now it's gotten kind of difficult. So if you start to consider that nanomaterials are being brought into the fold, they're much harder to detect in the environment. So you can't just keep testing suspicious liquids or things that you sample with a mass spectrometer. And they also don't always hold up under the conditions that you're going to do that analysis. So a lot of these molecules are really fragile and quite reactive, and that's why they're quite good at binding to acetylcholinesterase in the first place. So whilst this can be accurate, it's often not fast and it requires some Henderson instruments. So the community, being the dyers and fluorophores community, start to make little sensors and things that are able to respond to these quickly visually. But they also suffer a lot of limitations and often give false positives to things that are commonly found in the environment as well. So they're not very reliable. Then the enzyme folk are like, well, these things bind to enzymes really well, and have also engineered enzymes that break them down really fast, or could be like dosed out or sprayed or applied as foams. But one of the concerns was that there were all these poisoning events occurring, and the community didn't necessarily know how they were persisting in the environment so long, or how they're being delivered, or where they're coming from, or who's making them, because it's all meant to be controlled under the Chemical Weapon Convention. But if you're going to do nefarious things and kill people, you don't really have much regard for the rules. And certain areas of the world seem to have continued these development programs and are constantly developing new nerve agents. So some of you might be aware of these cases of Alexei Navalny and Sergei and Yulia Skripal being poisoned in the UK. One of them was administered using a perfume bottle. Someone later also collected that perfume bottle from a bin and then got poisoned as well. There are interventions that can take place. You can have like an antidote applied, but it has to be very quick, and it's a horrible recovery anyway, and obviously your health is generally quite impaired for some time. Once these nerve agents do bind to your acetylcholinesterase, the turnover of that enzyme is a really long period of time. So interestingly, it's hard to look up some of these concerns because they don't enter the general literature. So you've got to get on like weird platforms to try and find investigative journalism and reports. And one of that was intercepting reports associated with Russian companies that had shelled out from a previous weapons development plant after the oversight of the Chemical Weapons Convention, where individuals went off to make like sports nutrition drinks, or nutraceuticals or fertilizers. And they found that there was still quite a ring of interaction between these people, and they weren't very careful with their proofreading. So things like this would be found in their company reports, packaged within a veneer of another substance, allowing obfuscation and delayed onset of poison. So it seemed like they were still kind of messing about, and that was a concern to the intelligence community. So I was like, well, here's some stuff you might want to know about different nanoparticles. Here are some of the ways that they might be implemented. And then what might we be able to do to interfere with that, or disrupt them and still be able to test for the presence of nerve agents in a sort of front foot forward sort of way. So if any of these were to be successfully applied, they might conceal from interacting with sensors that rely on reactionary response, the fluorophores. They would have enhanced stability because you can control their local environment. They're more easily transmitted and you can get them through security much more easily, and very small amounts can be targeted towards people. So the overarching sort of illustration or graphic at the time was that we would encapsulate a surrogate agent, diisopropyl fluorophosphate, and determine whether we were able to detect it with a reactionary dye. So I was using a Bo-Dippy scaffold, a particular fluorochrome, and that's associated with a difference in colour upon binding to a reactionary head group. Unfortunately, and what I didn't know at the time, was that this field is plagued with issues with that particular cyclisation reaction. That particular cyclisation, whilst it looks really effective when you put any agent against it, it also gives the same colour response to a lot of these fluorophores if you put vinegar on it. So DFP, or other agents in the presence of glass or environmentally or in the presence of water, can decompose to produce hydrofluoric acid. That hydrofluoric acid kicks off other decompositions, and that generates a lot of the things that are going to give you false positives anyway. So often in application, some of these approaches aren't going to work. So a large amount of what I've been doing during this time is developing like strategies to sustain detection under different conditions, or working out different matrices to preserve a sample or screen against false positives. So during this time, I was halfway into this fellowship, I moved to Murdoch because UniSA was going into a merger, and I was like, this is not looking great. And I decided that I just didn't want to keep postdocing and trying to find obscure things. So I moved my fellowship to Murdoch and took a lecturing position there. And Murdoch is a very different sort of chemistry environment to where I'd come from. So where I'd come from was very biomedically orientated. It was very clinical health sciences, and the chemists were a very small contingent of that, but everything was generally biomedical and health orientated or biotech orientated. Whereas at Murdoch, the existing infrastructure was more minerals research, solutions chemistry, small ionic species, stuff like that. So a lot of the equipment is quite different. So since moving to Murdoch, I've continued the intelligence work, but have sort of pivoted into some other avenues. And one of those avenues became chemical ecology and toxicology. So I've linked in quite quickly with the forensics department, which is more sort of established at Murdoch for looking at some of the things that I might want to test for. And I'd been sort of carrying on some work that I'd been doing over in South Australia, which was the cat lures that I'm doing with Ashland, or cat and fox. And I came over here, and there's a group that want to look at natural compounds in plants that attract cats and for protection against invasive predators. So if you can modify their behavior or lure them into traps or whatever. So since being here, I've been here just over a year now. I've sort of, again, been faced with reinventing myself and doing some different things. So I've started doing some chemical ecology and analytical chemistry to support other research teams at Murdoch, and that's brought me into some really cool collaborative networks. It landed me my first PhD student that I'll have as primary supervisor, Juliet here, who was convinced to join me by Yvonne there, who is from forensics with a biology background, and joined me to do some sort of encapsulation chemistry. So this is the origins of a team. I asked Yvonne today if they had some professional photos that I could use for the presentation, and he's like, well, we've got one together, and he assured me that this was the safer one. So, and they're wearing Murdoch red, which is nice. So that's where I'm at now, and I'm sure there'll be a lot of other bizarre adventures moving forward. Thanks.
Well, thank you very much. Yeah, it sounds like it's been quite diverse. I enjoyed listening to your story. Does anyone have any questions? No?
very nice talk, very diverse talk. And I have a question regarding your first topic. With the different sugars, how big is the impact of the sugar on the uptake in the cells? So
Well,
if you compare glucose with mannose, for example,
yeah.
does it have the same ratio of uptake, or is this different?
It's different, but the uptake is largely governed by the lipophilicity of the complex, not necessarily the attachment of the sugar. So in an ideal world, it would have been assumed that cellular uptake was correlated to the metabolic activity of the sugar. That wasn't the case. So largely speaking, it's still driven by the fact that it's attached to a massive lipophilic complex. And the impact of the sugar was actually relatively minimal. So mannose actually looks like, by femtograms per cell, to be less than rhenium glucose. But if you use the control alkyne, it just floods into the cell. So it could be argued that potentially there's a contribution to active cellular uptake of the glucose, because it's a more commonly expressed avenue for uptake. But at the same time, it's largely speaking related to the lipophilicity, with minor contributions to possible active cellular uptake. The problem was that when we tried to do reduced temperature assays, the cells were just not tolerating it. And when we started using different inhibitors as well, it was just going to pot, and it didn't seem to meaningfully discriminate whether it was a contributing active process or not. And also, it didn't really vary that much by cell type. So we looked at H9C2 cardiomyoblasts, which should have massive cellular uptake by glucose in particular, which in this case was these. But we also looked at it in the prostate cells, and it was just nonsense. So I think it's just the fact that if you take a small biomolecule and you put it on a relatively large complex, then often the complex dominates the cellular uptake.
I've got a question for you.
Yeah.
So I like the way how you moved into the the neurotoxins and all that, and you said like trying to work out the best way to detect them. So what is the best and fastest way to detect them currently?
So currently they've got some it's like field-ready devices. So they'll use Raman and some field site mass spectrometers. But they also use these colorimetric kits. So it's like a drug typing. We do that as a prac. So you might be able to work things out into class, but they can work it out quickly. Is it potentially a threat? And if it's potentially a threat, then teams come in with a lot more PPE, and then they sample and they go elsewise. One of the problems that is faced is because these degrade so quickly in the environment. If there was a poisoning incident, forensically, you also want to be able to preserve that evidence. And in the instance that some of these some of these compounds that are being generated are massively structurally diverse, in terms of the Novichok series, one of the issues now is that because those side chains are so diverse in what keeps being detected, it's hard to trace that back to a source or who made them. So they are also particularly labile, and they decompose really quickly, and then you just have a phosphate or something and some stuff that you wouldn't necessarily correlate to the nerve agent. So it becomes kind of a challenge.
So it's a case of trying to get it as fast as you can,
Yeah.
so that you can actually try and pinpoint someone. Or is it like, if you're in the game of making these stuff, trying to make it one that deteriorates quickly.
Yeah, so a lot of them are kind of like viscous oils that are also somewhat volatile.
Yeah.
So like nerve gas, some of them are more like aerosolized sprays. That's probably another way to consider it. And if you were working with neat substances, then it's probably not as much of a challenge to determine whether they're present or whether they were used. But if they're minute quantities that are reliably stable and delivered in the case that nanoencapsulation might have been employed, then you've got an enhanced threat, but also more of a challenge detecting it. So that's the concern from the national security perspective. Yeah.
Yeah, it seems interesting and challenging at the same time.
Yeah.
So with your mineral flotation, is it nickel, was it?
Copper.
Copper mat?
Yeah.
If you use infrared, FTIR, you might be able to get a ratio of mineral to surfactant.
Yeah.
Optimise that and...
So that is actually something we had considered, but one of the problems is that the variance in the frother composition and what you buy in and batch to batch is so hugely variable that unless you have one distinct representative species in that mixture, it's kind of hard to work out because it gets sort of swamped.
Yeah, well, infrared will be able to tell you the concentrated surfactant, what you've actually got in there.
That's true. I think one of the other concerns that we had at the time is that it's so dynamic in that you might, over time, in a flotation bath, you might have an accumulation of certain frothers and you're losing others. So like the ratio changes over time as well. So it becomes kind of hard. But possibly something we could go back and look at. Yeah.
Any other questions? No? Well, if not, let's thank Todd once again. We'd like to present you with
Thank you.
for coming to talk to us today. So thank you very much.
Thank you very much. Cheers.